Hi, my name is Hayden Cecil, and this project is titled U.S. Government. Don't you, Carriot, be selfish. It's a democracy. Catherine Berkman and Tomas Garces Botero helped me to prepare this presentation and work from Mr. Bryan's 5th and 7th period AP Bio classes from the 2017-2018 school year. To make the following presentation more understandable, the following analogies will be used. Proteins will represent information, lipids will represent resources, nucleic acids will represent laws, and carbohydrates will represent money. The structure of the nucleus is that nuclear pores allow passage of RNA through the nuclear envelope. The double membrane separates the nucleus from the cytoplasm, and the nucleolus is at the center of the nucleus. The description of the nucleolus is on the next slide. The function of the nucleus is to protect the nucleolus and allow for protein synthesis by keeping DNA contained in the nucleolus and allowing RNA to pass into the cytoplasm. We felt that the nucleus would best be represented by the Secret Service because, just as the nucleus surrounds and protects the nucleolus, the Secret Service surrounds and protects the president. The structure of the nucleolus is that proteins and RNA create the mass in the center of the nucleus, which takes up roughly 25% of its space. The function of the nucleolus is to produce mRNA through the process of transcription, rRNA, which is part of ribosomes, and other kinds of RNA with the help of RNA polymerase. Also, it contains genetic material in the form of chromosomes of DNA. We felt that the nucleolus would best be represented by the president, who is currently Donald Trump, because as the nucleolus holds all the cell's genetic information and essentially runs the cell's processes, the president is in charge of all U.S. information and releases instructions on how issues will be addressed. The structure of the smooth endoplasmic reticulum is that it is made of folded tube-like structures around the rough ER, which surrounds the nucleus of the cell. The function of the smooth endoplasmic reticulum is that it creates and stores lipids, metabolizes carbohydrates, and processes toxins. We felt that the smooth endoplasmic reticulum would best be represented by the U.S. Department of the Interior because the same way the smooth endoplasmic reticulum stores lipids and steroids, the U.S. Department of the Interior safeguards and stores U.S. natural resources. The structure of the rough endoplasmic reticulum is that it is made of folded tube-like structures covered in ribosomes that are arranged around the nucleus. The function of the rough endoplasmic reticulum is that it is involved in the synthesis of amino acids and proteins and that it packages proteins and sends them to the Golgi apparatus for their distribution. We felt that the rough endoplasmic reticulum would best be represented by the U.S. Department of Education because, just as the rough endoplasmic reticulum creates amino acids and proteins, the U.S. Department of Education creates the curriculum saying what information is taught. The structure of the Golgi apparatus is that it is a series of flattened stacked pouches called cisternae. The function of the Golgi apparatus is that it packages proteins and lipids into vesicles and distributes them to either the cytoplasm, the cellular membrane, or outside of the cell. We felt that the Golgi apparatus would best be represented by the U.S. Postal Service because the Golgi apparatus receives and distributes proteins and lipids in the same way the Postal Service receives and distributes the letters and packages from and to the American people. The structure of the mitochondria is that the outer membrane contains an inner folded membrane which forms the layers of the cristae. A liquid is contained within the cristae called the matrix. Throughout the matrix, there are ribosomes and ATP synthase particles. The function of the mitochondria is to convert molecules of glucose into usable ATP energy by the ATP-ADP cycle. We felt that the mitochondria would best be represented by the U.S. economy because, just as the mitochondria converts glucose into usable energy, the U.S. economy converts money into jobs. The structure of the chloroplast is that stroma, the liquid in which the granules sit, is where the Calvin cycle or light-independent reactions occur. A granum is a stack of thylakoids, and thylakoids are fluid-filled discs in which light-dependent reactions occur due to the presence of chlorophyll here. The chloroplasts are surrounded by a double membrane. The function of the chloroplasts are to convert solar energy into glucose through light-dependent reactions in the Calvin cycle. Light-dependent reactions convert light and water into oxygen, and then the Calvin cycle converts carbon dioxide into sugar. We felt that the chloroplast would best be represented by the U.S. Mint because the chloroplasts create glucose, which later will be needed to create ATP, like how the U.S. Mint creates money, which later will be used to purchase products and services. The structure of the vacuole is that they are membrane-bound sacs for storage that are extremely large in plant cells and much smaller in animal cells. The function of the vacuoles are that they serve as storage for the cell's water, waste, and carbohydrates. We felt that the vacuoles would be best represented by the Federal Reserve because, as the vacuoles save water supplies and carbohydrates, the U.S. Federal Reserve Bank stores and protects American money and gold on which the country relies. The structure of the lysosomes is that they are surrounded by a phospholipid bilayer membrane, they are filled with a mixture of digestive enzymes, and that they are generally free-floating. The function of the lysosomes is that they contain digestive enzymes which break down enveloped bacteria, old organelles, waste, and food particles. We felt that the lysosomes would best be represented by the Supreme Court because, the same way the lysosomes remove non-functioning organelles and remove toxins, the Supreme Court chooses which laws are no longer valid and rules against criminals harming the U.S. The structure of the cytoskeleton is that it is made up of three components: microtubules, which are composed of alpha and beta tubulin; microfilaments, which are groups of thin fibers that form the cortical cytoskeleton inside the cell membrane; and intermediate filaments, which are made up of a group of related proteins with similar structural characteristics. The function of the cytoskeleton is that microtubules provide structure for cells and are the routes by which organelles move throughout the cell. Microfilaments act as a skeleton for the cell and create shape and structure for the cell, and intermediate filaments provide support based on the type of cell. We felt that the cytoskeleton would best be represented by the American people because, just as the cytoskeleton provides a structure for the cell, the American people provide the basic structure for our democracy and decide the form of all parts of our country. The structure of the centrioles is that they're cylinders composed of groups of microtubule triplets, and they're usually found in pairs. The function of the centrioles is that they release spindle fibers which aid in the division of chromosomes during anaphase of mitosis. We felt that the centrioles would best be represented by the United States Agency for International Development because, as the centrioles help with the division of the cell, the United States Agency for International Development spreads divisions of the U.S. throughout the world, mainly through the creation of U.S. embassies in foreign countries. The structure of the cell membrane is that it has a phospholipid bilayer, which is made up of a double layer of phospholipids with protein channels throughout. The hydrophilic head and hydrophobic tail of the lipids allow hydrophobic molecules to pass through. The function of the cell membrane is that it separates the intracellular environment from the extracellular environment, and that protein channels through it provide access for larger molecules, polar particles, and ions sent to the cell, such as carbohydrates. We felt that the cell membrane would best be represented by U.S. Customs because, as the cell membrane determines what molecules can leave and enter the cell in order to maintain homeostasis, the U.S. Customs and Border Protection determines who is allowed to leave and enter the country to keep it safe and stable. We hope this intracellular chip was extraordinary, and we tried to make it more extraordinary by giving you this great picture at the bottom of this slide. Thanks for watching, and I hope you learned something.